This is the antimicrobial agent susceptibility and resistance tutorial presented by the SchrDbb. We reference Lippincott Illustrated Review of Microbiology by Cynthia Cornilesson, Bruce Fisher, Richard Harvey. So we are going to visualize antimicrobial agents here. So these are the bar chart showing most commonly used drug families. So here is shown here. So we start with penicillins. You have cephalosporins, you have tetracyclines, aminoglycosides, macrolides, fluoroquinolones, and you have others. Now we start with penicillins. You need to know that penicillins are beta-lactam microorganisms that are names after the beta-lactam ring essential for the activity, and penicillins selectively interfere with the synthesis of bacterial cell wall. So that is for penicillins. And now we need to know a structure not found in mammalian cells. Penicillins are inactive against organisms that develop that are devoid in peptidoglycan cell wall. Examples: mycoplasma, protozoa, fungi, and viruses. So all those that are devoid of the peptidoglycan stains and which are mostly gram-negative are not going to be having a therapeutic effect based on the penicillins. So these are the different microorganisms that penicillins can work on. We have gram-positive cocci like Streptococcus pneumoniae, Streptococcus pyogenes. Streptococcus pneumoniae is a group B Streptococcus. Streptococcus pyogenes is a group A Streptococcus. Streptococcus viridans group. So all that can be the cocci where it can act on. Is it clear? So you can have also resistant strains which are increasingly seen. The second, we have gram-positive bacilli. In the case of Bacillus anthracis and in the case of Corynebacterium diphtheriae, can have penicillin effect. The next, we have gram-negative cocci like Neisseria meningitis. We have penicillin that act against it. We have anaerobic organism like Clostridium perfringens that penicillin can act against it, and we have spirochetes like Treponema pallidum and Treponema perteneri, where like in the case of Hoi and syphilis. Now the next one is cephalosporin. Cephalosporin is seen in another beta-lactam antibiotic that is going to be closely related to both the structure and function of penicillins. Is it clear? Cephalosporin have the same mode of action like penicillin, still acting on the peptidoglycan ring. Is it clear? But they are more resistant to the inactivation by the beta-lactamase enzyme. You need to know that penicillins are going to be affected by beta-lactamase enzymes. Why cephalosporin are now more resistant to inactivation with the beta-lactamase producing microorganism. Is it clear? Now we have different types of penicillins. We have first generation, second generation, third generation, and even fourth generation penicillins. Example of penicillins first generation: we have cefazolin, we have cefadroxil, and we have cefalexin. So the microorganisms which can be used. So the microorganisms that can be targeted by the first generation cephalosporin: you have Staphylococcus aureus, you have Staphylococcus epidermidis, you have Streptococcus pneumoniae, you have Streptococcus pyogenes, you have anaerobic streptococci. Now the gram-negative drugs that can be targeted by first generation: you have Escherichia coli, Klebsiella pneumoniae, and Proteus mirabilis. Is it clear? So example of cases which are methicillin resistant Staphylococcus aureus are resistant to these cephalosporins. Now the second generation cephalosporin includes cefuroxime and cefuroxime acetyl. Now example of organisms that are susceptible to it: you have Staphylococcus aureus, Streptococcus pyogenes, Streptococcus pneumoniae, and anaerobic streptococci. Gram-negative cocci which are susceptible to it: we have Neisseria gonorrhea, and gram-negative rods susceptible to it: we have Enterobacter aerogenes, you have Escherichia coli, Haemophilus influenzae, Klebsiella pneumoniae, and Proteus mirabilis. Anaerobic organism, you need to know that what cefazolin and cefotetan have anaerobic coverage also. Now the third generation cephalosporin, it consists of cefizim, cefotaxime, ceftazidime, and ceftriaxone. So the gram-positive, we have Streptococcus pneumoniae, Streptococcus pyogenes, and anaerobic streptococci. The gram-negative, we have Neisseria gonorrhoeae. We have gram-negative rods such as Enterobacter, Aerogenes, Escherichia coli, Haemophilus influenzae, Klebsiella pneumoniae, Proteus mirabilis, and Pseudomonas aeruginosa. But now Pseudomonas aeruginosa is only for peculiar cases like ceftazidime. Now, in other cases, summary of therapeutic application of tetracyclines. For tetracyclines, you need to know that in gram-positive bacilli, we have Bacillus anthracis. Gram-negative bacilli, we have Brucella species, we have Vibrio cholerae, we have Yersinia pestis. Is it clear? So a tetracycline plus a gentamicin is going to be used very effectively for this microorganism. So anaerobic organisms, you have Clostridium perfringens, we have Clostridium tetani that can be used here, Spirochetes, Borrelia, Bigdopharia, and Leptospira, interrogans, Mycoplasma pneumoniae, Chlamydia, all the microorganisms and others, even Rickettsia, all the microorganisms that were atypical, you can use tetracycline. Now, summary for therapeutic aminoglycosides. You need to know that what therapeutic aminoglycosides, you have cases of gram-positive cocci like Enterococcus species, where you can use gentamicin plus ampicillin. You have Streptococcus agalactiae, where you can use gentamicin plus ampicillin. Now, gram-negative, in the case of gram-negative rods, you can use Brucella species, gentamicin and doxycycline. Francisella tularensis, you use gentamicin. Klebsiella species, you have Yersinia pestis, you have you have Pseudomonas aeruginosa. So all these are the microorganisms you can visualize, you can use here. Now, in case of summary of therapeutic application of macrolides, so when you want to use macrolide, you can use for Staphylococcus, for gram-positive cocci like Staphylococcus aureus, Streptococcus pyogenes, Streptococcus pneumoniae. You can use for gram-positive bacilli like Corynebacterium diphtheriae, gram-negative cocci like Moraxella catarrhalis, like gram-negative rods like these cases. You can use for Spirochetes, even for Mycoplasma atypicals like Mycoplasma pneumoniae, Uroplasma ureaticum, Chlamydia like Chlamydia pneumoniae, Chlamydia cysticercae, and all that. So these are now the therapeutic use for the fluoroquinolones. Fluoroquinolones are mostly used for gram-negative rods. You see that yes, a high coverage for gram-negative rods like Enterobacter species, Escherichia coli, Haemophilus influenzae, Klebsiella pneumoniae, and all that. And the gram-positive bacilli is only Bacillus anthracis. Is it clear? Now, for the antimicrobial coverage of imipenem, imipenem is one of the highest drugs inside the microbiology scale. Is it clear? And that's why it has the highest coverage. Cover gram-positive cocci, cover gram-positive bacilli, cover gram-negative cocci, cover gram-negative rods, cover anaerobics, and it covers others. It has a very great list. Is it clear? That's why this drug, which is imipenem, is going to be the most used of all, and it's the best use of all. So now we have drugs. So this is a schematic representation of drug resistance. So drug resistance is just because the drugs can be pushed out of cells from the intracellular into the extracellular milieu. Is it clear? So we have different forms of drug resistance. The first one is spontaneous mutation of the DNA. So this one, genetic alteration, is into drug resistance. The next one is DNA transfer of the drug resistance. So when one microorganism has gotten the drug resistance, it can transfer its plasmid to another microorganism, such that the other microorganism has also a factor for drug resistance. So those are the genetic alteration leading to drug resistance. The first one is spontaneous mutation of DNA, and the second one is a DNA transfer. Now we have altered expression of proteins in drug resistance organisms. So when you have altered expression of protein in drug resistant organisms, you have modification of target sites, you have decreased accumulation, you have enzymatic inactivation. The first one is modification of target site. Is it clear? So when the target site of the microorganism is modified, you can have one form of drug resistance. You have decreased accumulation, means that what in case of decreased accumulation, you have the expulsion of the interest from the intracellular to extracellular membrane, so that the antibiotics do not accumulate inside the cell again. And then the third one is the enzymatic inactivation, where certain enzymes such as beta-lactamase are used to inactivate the beta-lactam rings of the penicillin. So as an example. now to what are the agents now which are used to treat viral infections? Now you need to know that the medically important viruses are organized in similar groups based on the nature of the genomes. The first medically important is single-stranded non-envelope. You have double-stranded non-envelope and you have double-stranded envelope. So all this, when you see red, it means that it is RNA. Now the next one, when you see in blue, it means that it's DNA. So this is single-stranded. No, the one in red is DNA and the one in blue is RNA. So single-stranded positive strand equals a helical non-envelope. Single-stranded positive strand equals a helical and envelope. Single-stranded negative strand helical and envelope, and all these are involved with the medically related microorganism. So now we have different antiviral agents which can be used. Is it clear? For influenza virus type A and type B. So these are, for let's start with double-stranded envelope. So when you have double-stranded envelope DNA. So for double-stranded envelope DNA, we have hepatitis B. In case of hepatitis B, you can use all these antiviral drugs. You have Adefovir, anti-cavi, and all that with lamivudine, tenofovir. Still in herpes simplex virus, you can use acyclovir, famciclovir, ganciclovir, valaciclovir. In case of human cytomegalovirus, you can use cidofovir, ganciclovir. So it's the only one which can use. You can also use foscarnet. In case of varicella zoster, you can use acyclovir, famciclovir, valaciclovir. Now in case of single-stranded positive strand icosahedral or helical and helical or envelope, you can see influenza virus type A and type 2, type B. You can use zanamivir, you can use rabavirin, and you can use oseltamivir. So you have hepatitis C. Now hepatitis C is single-stranded, negative strand, helical, and envelope. It is single-stranded. The number of strands, it is positive or negative. Here in this case it is negative. The shape is helical and it is envelope. So now when you have this example of this microorganism, we have hepatitis C, and you treat hepatitis C with ribavirin, or you treat with pegin interferon. You can use interferons mostly here. Now in case of immunodeficiency virus, because immunodeficiency virus is an RNA virus which has a negative strand, and it's envelope too. So you have HIV virus 1 and 2. You need to know that the microorganism, the antiviral drug which are used in HIV 1 and 2 are divided into nucleosides and nucleotides analog to reverse transcriptase inhibitor. Is it clear? So it is one for one drug that is used. So that's one is called nucleoside antiviral drugs. You have non-nucleoside reverse transcriptase inhibitors. You have first one, which is nucleoside and nucleotide reverse transcriptase inhibitor. You have non-nucleoside reverse transcriptase inhibitor. You have the next one is protease inhibitor. The next one is fusion inhibitor, and the next one is integrase inhibitor. Because all these are contained inside the HIV virus. The first one is nucleoside nucleotide reverse transcriptase inhibitor. We have abacavir, you have didanosine, you have M3C-tabine, you have lamivudine, you have tenofovir, you have zidovudine. All these, even stavudine, they are inside nucleoside nucleotide reverse transcriptase inhibitor. Non-nucleoside reverse transcriptase inhibitors, now we have efavirenz, we have nevirapine. Now we have efavirenz and nevirapine. Protease inhibitor, we have the protease, we have indinavir, we have ritonavir, we have sakinavir, we have lopinavir. All the navirs are inside here, the protease inhibitor. The fusion inhibitor, we have the enfuvirtide fusion inhibitor, and the integrase inhibitor, we have the raltegravir. So all those are the microorganisms involved. All those are the anti-microbial agents involved here. So from here we say thanks for your kind attention. Please don't forget to like and subscribe for our channel. Thank you.